Sprechprobe 1, 2, 3, Soundcheck.
And shall it So good afternoon, everyone, and welcome back to our cell biology practical course online. And today we proceed with experiment number 2, and we will analyze results of subcellular fractionation, what we have performed yesterday. I will explain to you in the beginning some basic principles of the protein electrophoresis method SDS-PAGE, and then and later Western blotting. We will go into details of our practical book protocol, and in the end of each hour we will have a video tutorial with all the details as it was yesterday, that you can see everything in the videos. At the end of today's presentation, there will be also short presentation on the experiment number 3, WST cytotoxicity assay. So again, if you would have any questions or comments, please write everything to the chat. So before we start, let's refresh a little bit our memory what we have done yesterday. We have cells, we harvested cells with a scraping, with rubber policeman. We homogenized cells with the needle and the syringe. We centrifuge, homogenate to obtain supernatant, post-nuclear supernatant. And then at the last step, we used ultracentrifugation at 100,000 G for 1 hour to separate post-nuclear supernatant fraction into the two fractions: cytosol, supernatant after centrifugation, and total membranes of the cells, which was a pellet P100 after ultracentrifugation. So now what we are going to do is to analyze that with Western blotting and the marker analysis. And as we already have discussed, we will detect the transferrin receptor as a marker for the total membrane fraction, and tubulin as a marker of cytosol. I would like to mention that yesterday when we were watching all the video tutorial for this subcellular fractionation method, and I also in the introduction showed you examples and mentioned that this is a tricky method. And I realized when we were watching videos that probably you're the first students, the first group, and myself also first time that I could explain everything, this rather difficult method, explain everything into such details that I could show you all the steps, all the pellets, how it's important to do homogenization, how to hold or how the syringe, and so on and so on. So I think these videos will give you really unique chance if you come then later to the laboratory to perform real wet lab practicum, that you will know everything, how things, also difficult things and tricky things, how they have to be done. So, and then we have today these similar videos that you can see everything in details. Now, so we start thinking about the Western blotting and marker analysis. But before we go into this method, I would like to remind us that we finished yesterday with this graph and results of the Bradford assay examples I gave to you, and I ask you this question: what volume of post-nuclear supernatants, 3 different samples prepared by students last year, makes 30 micrograms of protein for Western blotting? I would like to ask you, I ask you to answer this question into the chat now. I'll try to explain you why it is important. So basically, we are not yet in the stage to start electrophoresis. We will talk a little bit about that, and then we will start it later. But just imagine that we have not sufficient amount. Let's aim for these 30 micrograms. But if you don't have a 30, well, we can use 20 or so. But it's really good to know where we are, because basically if you have not sufficient amount of protein, if at all, for instance, something didn't work, then there is no reason to proceed with the analysis, because we don't have a protein. This was the reason why we performed the Bradford assay yesterday, to determine the protein total amounts. And I hope that we would need 1, 2, or a few answers from a few of several of you, that we know, that we see the numbers. So I hope you have access to chat and give me a few answers, that we see how far are we from expectation, and hopefully we are in a good level. Just imagine that we are not. So what do we do? We should go back to the original, right? And now online it's easy. I would just enlarge the dish. So this is 10 centimeter dish. I would extend it to the 15 centimeter dish, and we will start experiment with 15 centimeter dish of cell culture of HeLa. And someone who is good in mathematics would immediately say, well, if you take, instead of 10 centimeter dish, we would take 15, we would get 3 times more cells from this dish. So we could scale up our experiment. This is easy done now for me. I just would make the dish bigger, and then we are ready. In real life, it would take a bit longer time, right? Because we would have to start with a cell culture. Cells should grow, so they should grow from the stalk after trypsinization. It takes a few days, and then we need a 15 centimeter dish, and then we should start again our homogenization procedure, and basically it would take us another 1 week or so. So let's hope we have enough sample, and there are answers in the chat. So PNS1 12, PNS2 14, PNS3 15 microliters. That's Elias. Moritz 6 microliters, 7 microliters, 7 microliters, and Yasmin 12, 14, 15 microliters. So we have already different answers, right? And maybe let's, I think we should have enough time today. Let's spend another minute to clarify and find out who is right. At the moment, I don't know. Let's do this together. And since you have difficulties, I will just guide you through such a data and show you that one can do this, I hope, fast and rather easy without computer and the even Excel program. So we have raw values, for instance, for sample PNS1. This is 0.81, 0.84. So the average would be 0.83 or so. We go to the optical densities, which we have measured for the standard curve, and we stop in the position of, it was 0.83, somewhere here. We go to the right, and we come to the standard curve, which would be close to this actually concentration of BSA. So we basically go down in this value, and what do we see? We see 5. So 5 means 5 micrograms of protein. It was 5 micrograms of protein in the sample which we have measured. Let's take a look how many microliters we took. So we took 2 microliters. That means 5 micrograms in 2 microliters. We divide 5 per 2, and we get concentration 2.5 micrograms per microliters of protein in PNS sample number 1. So if you have 2.5 and we need to load 30 micrograms, that would correspond to 12 microliters of the sample. Let's see. We have answers and correct answers from Elias and Yasmin, 12 microliters of the sample. So I hope the Moritz also now understand how to do this measuring, and for fun you can make these quantifications later, or this rough estimation for the other samples. Obviously the values are here, so you should take more a little bit, and the more for the sample number 3. So I hope now you all know how to quantify the protein with the Bradford assay, and also with the graphical method I showed you, very fast and easy way. And most important is we agreed that we load, not load, but we need something between 12 and it was 15 or so microliters of the sample. Now we come back to the question from yesterday. I think this was Catherine who asked yesterday, why do we collect 40 microliters? Now we know 40 microliters is collected for the Bradford assay. We use 4 microliters. 36 are left, and we need to load basically 12, so less than 20. We have sufficient amount of the aliquot, post-nuclear supernatants, collected. This is good. So we are now sure that we can proceed with our experiments, with analysis, and that's why we go to the next slide. Now I will, let's look at the protocol, so page number 15 in our protocol book, and let's look what do we need for equipment and materials for the SDS-PAGE, sodium dodecyl sulfate polyacrylamide gel electrophoresis. This is a full name of the method. Yeah, we need a gel electrophoresis unit. This is something what we use in the laboratory in the Praktikum room, comes from the one company, but there are many different, and we will see it in the future in different laboratories. So we will use such a unit. We need centrifuge, thermoblock to prepare sample. We need gel cassettes, the comps to basically plan our experiments, like for instance 10 well comps, 1 millimeter thickness we are using in the Praktikum, but the number of well in the comp could be different. Let's say for the same electrophoresis, which we have the unit, it's small mini electrophoresis system. This could be 8 well. This could be also 15 well comp, right? So basically we plan in advance, and also we can change it just before we start experiment in the case, for instance, if the concentration of protein is low and we need to load bigger volumes. Second thing is that here we use 1 millimeter thickness for the comb, but of course corresponding cassette should have also 1 millimeter thickness, and if we need to load more sample, for instance, we can use instead 1.5 millimeter thickness or 0.75 just as an examples. Right, we will see we use a special gel loading tips. They are convenient to load the samples in the gels, the EPIs, the tubes, and again plastic transfer pipettes. Basically, this is the list of equipment. Second, we have a solution. So the buffers, which several of them, we will prepare ourselves during the Praktikum. So we use sample buffer, for instance 4x SDS sample buffer, and you see the composition. So it's buffered with trees, pH 6.8. It contains DTT and SDS and the glycerol and bromophenol blue. I will explain to you in the next slide all the components, how, what they do, why we use them in the sample buffer, and how we prepare sample. Maybe now in advance I will ask you one question. You see what this abbreviation SDS, so sodium dodecyl sulfate, and I will ask you a question and expect your answers in the chat. What SDS does to the proteins? What is the function? And if you know this answer, please write it to the chat. Protogel, this is the mixture of acrylamide and bisacrylamide, methylene bisacrylamide. It's a solution of 30% and 0.8%, two components. Some time ago we had this as a powders, but the powders are extremely toxic, so they're neurotoxic, and since many years we use already this pre-mixed mixture of these two components just because they are toxic. And in any case, with the solutions we should work carefully. When the gel is polymerized, then it's absolutely safe, but when it is in solution we should work very carefully with this. So these are two components which make the gel. They polymerize and make the gel, polyacrylamide gel, right? So we use a buffer, so this we will prepare ourselves, resolving gel buffer. You see the molarity of it, and the pH is 8.8. The stacking gel buffer, which you use to prepare stacking gel, is a 3HCl buffer, pH 6.8. We use a stocks, 20% SDS, and two components, stock of ammonium persulfate and also TENET. So maybe you know already that these two components are used, APS, as initiator of the polymerization reaction, and the TAMET is used as the catalysts of the polymerization reaction. And this is the composition of electrophoresis buffer we will also prepare ourselves. So this is tris, glycine, and SDS. So this is basically what we will have ready before we start electrophoresis. And now I will explain you step by step all the components and the function of them. And as you see in the sample preparation step, very important for the method, we add to our sample, which we have already prepared, we add the SDS sample buffer. Usually it's something between 1x to 5x stock, and the composition of 1x sample buffer is shown here. So it's 68 millimolar 3-HCl buffer, pH 6.8. It contains also 100 millimolar DTT, 2% SDS, glycerol, bromphenol blue. So actually on this moment I ask you, and I on purpose did it in advance, that we have more time and that you think and answer into the chat what SDS does, and then I will check now chat if we have some answers. So Moritz answered, it unfolds proteins and gives them negative charge. Elena, SDS is for denaturing proteins. Tommaso, SDS neutralizes the charge. Isabel, charges protein negatively. Elias, it denatures the proteins to avoid unwanted secondary structures. I'm very happy to read all the answers are absolutely correct. They cover all the different, so to say, details how SDS functions, and you all basically covered all these details in your answers very well. Happy to read that, and we will come to this in the next slide immediately. So basically, just remember the tris buffer pH, DTT. We will come to the next slide. SDS we already know. The glycerol is added to make our final samples to load just a bit more dense, so heavy, that we can nicely load the samples into the well. And bromphenol blue makes the mixture blue that we see contrast of the sample and also how we load it, and also that it will run with a front at the end that we see when it reaches the end of the gel. So basically, sample preparation is very fast and simple. We have the sample buffer. In the practicum, you will have it from me. And we mix it by vortexing. Sometimes it's necessary to make a pipetting ups and downs. And incubate 5 minutes at 95 degrees and centrifuge 5 minutes. So basically 10-15 minutes and we are ready with our samples. So now these, your all answers feed to my next slide. I wanted to show you the SDS, sodium dodecyl sulfate. So this is the structure of this detergent. Yeah, it's anionic detergent, obviously anionic because it has this sulfur group. It's negatively charged. It's also hydrophilic part of the detergent, right? And hydrophobic region is here on the red to the left, and some of you who remembers from school Latin, you can conclude that dodecyl means 12 carbon or methyl and actually groups here, because its undecan will be 11 and dodecan will be 12, right? So there are the 12 methylene groups, and this is the long, large hydrophobic region. These are structure of the SDS. And when we add our sample buffer to proteins, so the one component what we didn't discuss yet is the reducing agents, and what they do, they basically they reduce these SS bridges, and you might remember from previous courses that these SS bridges are made between cysteines, amino acid cysteine, right, in the proteins, and this is how it looks like when protein is folded and also has these SS bridges. We use reducing agents For instance, in our laboratory, the very common and standard one is DTT, DTO-trietol, but there are many other different one. Probably second common would be beta-mercaptoethanol. It's also very common, or recently, although quite many years already, some modifications of very strong reducing agents based on the phosphine, like TSAP, are used in the laboratory. They all do their job, so they cleave as bridges, and they reduce these groups, and keep, actually, since we have it in the sample buffer, it keeps in this reduced condition cysteine amino acids, so it helps unfolding proteins. Unfolding itself, as you have already mentioned, is done by SDS detergent during this 5-minute incubation at 95 degrees. We often call it boiling, although it is not really boiling. It was boiling before, that you can boil it in the water, or incubate 5 minutes in the boiled water. Now we use 95 degrees incubation, and then reaction, this unfolding, the reducing and unfolding occurs. And as you already, many of you mentioned in the chat, so what we get is unfolded protein, which look more or less like extended road, and when it is covered covered with the hydrophobic part of the SDS detergent, so what we have on the surface of such a long road, or actually we can even call this sausage, we have these negative charges of the sulfate sulfur groups, right? So all the proteins look very similar. That's why one of the comments which was online, that we mask differences between different proteins, right? So we have, let's say, roughly 20,000 proteins, or if you think about modification, it could easily come to 2 million proteins in the tissues, and then all they are unfolded and masked with SDS detergent. They all are negatively charged and look like this extended sausage. So what is very interesting, that the binding of SDS to the majority of proteins is in the rough ratio of 1.4 to 1, so in this case, grams of SDS to gram of protein, or milligram of SDS to milligram of protein. It's not exact, it's not like always like that. There are some quite many examples of different proteins that the binding is not exactly the same, but the major or general principle is like that the binding is in this roughly this ratio, and therefore all the proteins have the same charge to mass ratio, and what we can see in the gel and separate the proteins in a way that protein mobility is a function of molecular weight. So this is the SDS, the main function and the idea of this method, that we can separate protein based on their molecular weight in general. So this is the main or very important principle of the sample or an idea of sample preparation, as well as the main principle of SDS electrophoresis. What I would like to show to you in this slide is the second basic principle of this method we are going to use. I'm not saying second because it's less important than the first. It's just the two main things what I would like to introduce to you, that you understand why it is important. And as we discussed yesterday, and some of you, I believe it was Elias and Magdalena, probably they found that the first second paper was the paper we refer today. It's a Nature paper from 1970, published by Lemley and colleagues, and it describes this SDS-PAGE electrophoresis as a discontinuous pH buffer system electrophoresis. And now you remember we discussed yesterday Bradford protein assay, determination of protein concentration, as the third, the most cited paper. Now we come to the second most cited paper, which is Lemley, publication in 1970. And just for your, maybe some of you are curious to check, not necessarily now, but when you have time, also after our stream today, to check also what was or what is the first most cited paper in the scientific literature. I would ask you not to search it immediately now, because I want you to focus on something what I'm going to tell you now, and this is something important. That you understand the principle of this electrophoresis and the trick how to get final results of a good quality. And the conclusion, so this is basically the main conclusion from this publication is, and principle of the method is that focusing, here it is, I put it on the second line here, the focusing of the proteins actually happens before entering the separating gel, right? And this is something what often people do not remember or tend to forget, or sometimes do not understand. So this is the main principle of this method, that proteins are focused before they enter the resolving or separating gel. And if you will understand this main principle, I'm sure that in the future you'll get nice results of Western blotting analysis. So what was the trick? And now I'm going to introduce this and to explain this trick to you. And the trick was in the electrophoresis buffer. We still call it Lamley buffer, or the Lamley electrophoresis buffer. And this is the composition which was already mentioned in the previous slide. Is 25 millimolar trees, and the glycine, quite high concentration of glycine, and SDS. So SDS we have everywhere. So we have SDS in the buffers, we have it in the sample buffer, as we discussed already, and also in all the parts in the stacking, so upper concentrating gel and the second gel which is resolving or separating. So we have SDS everywhere. Now N3 is everywhere. So what is the difference? And the trick of the method is basically can be explained by this component which is glycine. As you know, it is amino acid. And this is very special and unique amino acid because of it's a weak amino acid, or a weak acid. And I would like to ask that you follow me in this scheme, the behavior or the charge actually of the glycine amino acids depending on the pH of the buffers. So let's start, for instance, in the low pH. We have a lot of H+, right? Low pH means acidic conditions, and this amino group contain or has the H+, that it's positively charged. There are a lot of the H+ in the solution. It's acid, acidic conditions, low pH, and also this carboxy group is neutralized. So basically the glycine in the acidic conditions would be positively charged, right? So when we remove H+, so we go more to the neutral pH. So neutral we call something around pH 7, as you know. So we have less H+, and we kind of have it, it goes away from the carboxy group, and it's negatively charged in the neutral pH. The charging of the amino group in this amino acid stays positive. So what do we have in the neutral conditions? It's called so-called Zwitter ion of the glycine. So it has no charge. The total charge is basically 0. When you go more to the basic conditions, so even less H+, right? We go to the right, so we don't have H+ anymore on the amino group, but carboxy group stays negatively charged, and it happens at high pH. And the glycine in these conditions is negatively charged. So this is basically the whole idea of the method, that there is a difference between the charge of the glycine in the neutral conditions and the basic conditions. So let's take a look why it is important. So what do we have? We have a 3-glycine as the electrophoresis buffer, right? So this, which is here described. This is our, let's say, upper chamber when we have a buffer. Then we come to the sample and the concentrating gel. pH of these two components is 6.8. Then we look at the resolving gel. This is 3's HCl buffer, pH 8.8, so it's basic, right? And again, we have at the other, and let's call it bottom, electrophoresis buffer chamber. It's again the same 3's glycine buffer. In general, as you see in this composition as well as in the total, there is no HCl. So what is important that we do not adjust pH of this buffer. So we don't even have to write here. It just, in the purpose of the explanation for today, I mentioned the pH of 3's glycine buffer, but we never adjust pH of this buffer, and maybe you know it already why, but if not, then I'll explain it to you now. So basically we have three, or actually draw the three situations, three conditions for this electrophoresis. So what we have before we start? We have 3's glycine buffer in the top. We have 3's HCl buffer pH 6.8 in the sample and this in the stacking gel. And as we already discussed here in the resolving gel, it is basic. This is before we start electrophoresis. So what happens when we click on the start and we have electrophoresis procedure started? Let's take a look. So as we agreed, already proteins are negatively charged, they start to move. pH 8.3, the glycine is negatively charged, it's also start to move. So everything starts moving. The chloride ions, they are always negatively charged. So these are simple. So HCl is a very strong acid, and the chloride ions are always negatively charged independently, basically, or in these conditions we use. They are negatively charged, and the chloride ions are very small. If you compare to the glycine, the chloride as the ion, these are small ions and they are negatively charged. They really run fast. So all the chloride ions from the stacking gel, they run first, the highest speed, and if that would be the competition. The proteins, they are different of size, but still we think about something like kilodaltons, right? So 10 kilodaltons, 20, 200 kilodaltons, and so on. So they start to move. They also move from this well, as we loaded it, they start to move, and they slowly start to move. They never can overtake the chloride. Chloride is small and fast. What happens to the glycines? They start moving, they are okay, they are negatively charged, they are not as fast as the chlorides, they are bigger, and then suddenly they enter, from the condition of 8.3, they enter stacking gel where conditions are 6.8. So what do we have in the condition of 6.8 pH? This is neutral. The glycine is not charged. It doesn't move. So the situation in the middle is actually the main principle of this electrophoresis, that the chloride ions move first and fast. The proteins move, but the glycine, in a certain moment, they basically start moving very slow. So all the power of the current we start, so this negative, the electrodes, all the power of the electric field come onto the proteins. They run, and they basically, during this run from the well to the resolving gel, they come in very, very narrow, very concentrated, and you can see this, or maybe you saw it already before. Doesn't really matter the volume you load. It could be very, like if you would say what you see could be 5, 6, 7 millimeters. They here will come to the very narrow, less than 1 millimeter band as this blue, bromphenoid blue. So this is the secret to success of the separation in electrophoresis, because the glycine cannot overtake the protein, proteins cannot overtake the chloride, and when they enter or they come to the resolving level here, they are all super concentrated. And then if you reach such the situation, then the next step will be most probably successful. What happens in the last step when the proteins enter or reach the resolving gel? As you see here, pH is 8.8. We have again back charged negatively and stronger than before. The glycine ions, they start to move. They are small, they are negatively charged, they overtake the proteins. The chloride ions are even already faster, and they are fastest of all the ions present in the system. So from the moment when the proteins enter the resolving gel, they start to be separated based on the principle which we discussed at the beginning, based on the size, so molecular weight, and we can separate large proteins, let's say something like 200 or 300 on the top, down to 5 kilodalton small proteins in the gel. So this is the second and the last phase of the protein separation, and we usually run electrophoresis down to the end of the gel when the bronchphenol blue reach the end, and this is what we see at separation, or what we expect, separation of the proteins based on the molecular weight. So maybe now, I hope you understand this second important principle and the focusing of proteins before the resolving gel. I would like to ask you a question. So what do you think? Would it be good idea or it's bad idea to prepare your upper concentrating gel in advance? Let's say you have experiment today and you made it yesterday. So is it a good idea or bad? And if it's bad idea, why you think it is bad idea to prepare your separating upper gel day before? And why maybe it is better to prepare when just before the running electrophoresis? So if you have your thoughts, ideas, please write it to the chat, and we will check it in a few minutes if what you think about this principle of the separation. Meanwhile, I will start slowly explaining to you the details of the practical protocol book. So these were before I gave you some general comments and the principle of the methods and the separation. And now we come to the details. What do we do for the sample preparation as it stands in our protocol book? So to our samples we add 4x SDS sample buffer, 2 postnuclear supernatant, and 2 cytosol aliquots, right? Mix well by vortexing. Should be everything clear here on this step. Then is to prepare 1x SDS sample buffer. From the stock you have 4x. I hope you will not have a problem with that. To prepare 1x, and we add this 1x buffer, 100 microliters of 1x SDS sample buffer to the P100 sample. So total membranes. You remember we have yesterday aspirated the supernatant from centrifugation, ultracentrifugation sample, and this is now staying on ice, so usually just a few minutes, as the P100 sample. To this sample we add 100 microliters of the 1x SDS sample buffer. So you might remember yesterday that we took 100 microliters of the postnuclear supernatant sample. So this is exactly the reason why we add 100 microliters of SDS sample buffer. So we basically have the starting sample, which was 100. We fractionate it into 2. Cytosol stays as it was. It's a liquid fluid in the supernatant, and we reconstitute the pellet back to 100 microliters like it was originally. So what is important now in this step is to resuspend the pellet thoroughly by pipetting. So this pellet of the P100 you need to resuspend with the tip, ups and downs, and vortex. So after vortexing and resuspending, we have all the samples ready, and we incubate samples 5 minutes at 95 degrees, centrifuge tubes 5 minutes, 13,000 G, so basically maximum in your tabletop centrifuge, and we prepare pre-stained molecular weight markers. So far we didn't discuss molecular weight markers, but important is now please don't mix the term markers with the markers which we are going to use as the markers of the different fractions, protein markers. These are molecular weight markers, and I will explain them to you in later today in examples how these markers, molecular weight, is basically ladder or the mixture of different molecular weight proteins of sizes we know. So now we are ready. Just a brief reminding, so you can compare the details of what we discussed, the general principle. Today we use 10% polyacrylamide gel as a resolving gel, and of course these can be taken basically almost any percent, depends on the range of molecular protein weight we are interested in to be detected in the Western blot, right? So we take a 10%, but we can take 7 or 15% gel, depending. If you want to see low molecular weight, we take higher percentage of the gel. Let's say if you want to see 10-15 kilodalton proteins, we take 15% gel, and if you want to see high molecular weight proteins, we take low percentage, let's say 7 or 8%. There are also there is a possibility to run so-called gradient polyacrylamide gel, for instance from 5 to 20%. So all these details probably you will see in the future, and it depends on the experiment you're going to do. So what we do, we basically mix the components as a buffer, water, protogel, SDS to get this 10% gel in the tube, and just before running electrophoresis, we add APS and TAMAT, as we already discussed. They are a catalyst and the initiator of polymerization. These are, after this everything is ready for the resolving gel, we cast the gel, and you will see it in the video. And this is just the composition of stacking gel. It's 4% gel. And how much we take the buffers, we will prepare it, or you will get it from us. Again, buffer, water, and protogel and the SDS. So basically, these are all the components we mix. And when we are ready, let's think. I will check in the chat if there are answers. So there is answer from Elias. No, from Moritz now, that maybe the pH gradient would blur due to diffusion of molecules if you prepare it too early. Absolutely correct. So if you prepare gels too much in advance, you can imagine, so basically let's go back 2 slides. So if you have these conditions and you have stacking gel which has a 3-HCl pH 6.8 and the resolving gel 8.8, so and if you keep it for too long, let's say, the difference is basically between the chloride ions, right? So they can easily diffuse, and you will not have a sharp differences between concentrating and the resolving gel. So it's always better to prepare it just before stacking gel, just before use. We will do this with you 30 minutes virtually now, and practically in the course 30 minutes before we start electrophoresis, that we have this effect of pH as good as possible. Of course, if you do this in advance, that basically the difference, as you have seen now, 4% acrylamide, and in the stacking gel, and 10 or even higher in the resolving gel, will be still important. The proteins will concentrate when they start entering the gel, but this will be just over half of the success, and the other half difference between the pH will be a little bit or significantly blurry, so you will not see such a sharp bands, and the future bands in the Western blot will also be not sharp. So thanks for this answer. Now we can proceed. We were here. The composition of the resolving and the stacking gel, we discussed that. So when we're ready, we assemble the gel in the running chamber. We fill the electrophoresis buffer. Now we load the samples which correspond to original, so volumes of 3 samples which correspond to 30 micrograms of the protein in post-nuclear supernatant. So you will see not more than 25 microliters. You will see that there is a certain physical limit of the well. So you cannot load more than certain volume. Yeah, it depends on the thickness, 1 or 1.5, for instance, thickness of the well and the gel, but 25 is something that would be maximum. If you try to load, let's say, 35 microliters, of course, you can come to the spilling over sample from the one well to the next well, and you will get some artifacts, some mistakes on the wrong result. So basically, this is the scheme I propose, how we load the markers. We will use, I will show you examples of the pre-stain markers, and we load, for instance, post-nuclear supernatant, cytosol, and the total membranes. That will be sample number 1, which we have already analyzed the concentration from one student, the second student, and the third student. Now you see all the 10 wells will be occupied in the 10-well comp, and 3 students. Usually this was our standard group. So 3 students in one group, so that means one gel would correspond to the one group in the Praktikum in the future. And when we are ready, we start electrophoresis at constant current 20 milliamperes per gel. Also constant current is always better to run, and we put a maximum voltage, for instance 170 for this system, which we will use in the Praktikum, and it will run until the blue dye, bromophenol blue, reach the end of the gel. So I think I tried from my side to explain you all the details on the slides, and giving and discussing with you some values and the numbers, so we are ready to watch the video tutorial. I will start now. What you see is the first page, what we discussed already from the protocol book, and how I prepare resolving gel. So I have mixed already the components which were on the table, and now I add, just before use, I add APS and TEMET. So this was APS, initiator of polymerization, of the reaction between the acrylamide and bisacrylamide. So this is a radical reaction, and the radicals come from initiator, from APS. And the second is a catalysator, so the TEMET, and we add, so here we see this is 5 microliters of the TEMET. We add to the tube according to the protocol. We mix it well. So now I add it to the tube where all the components are in already. Add it, close it, and mix it well. So now we are ready to fill this cassette. So this is a plastic cassette for single use. It's commercial, but for instance in our laboratory we use self-made glass cassettes. So for you we use just more convenient the plastic cassette, where you fill the mixture of the polyacrylamide and all the components with the plastic transfer pipette. So please do this carefully and add it to the level, as you see, something like 1.5 millimeters from the top, more or less on this level, what you see horizontal here, a little bit higher, and we overlay with the water. In the laboratory, we use different solutions, like for instance, isopropanol or water-saturated butanol to overlay. You see the water also comes on the top. So maybe you see it on your monitors, or actually that water comes here on the top of acrylamide. And this is the reason that we want to have sharp top of the acrylamide gel, and water works quite nicely. but also other alcohols can be used to make nice ending, or the beginning, so to say, if you think about the protein entering the gel from stacking to resolving. We repeat the same with the stacking gel. We let this resolving gel to polymerize for 1 hour at least. So the polymerization of resolving gel, the longer the better. And at least 1 hour after polymerization, we prepare stacking gel in the same pipetting. So the all components are already in the tube which I have in my hands, and I add APS and TEMET, and I mix it well. You can always use the vortex, it's all on your bench. And I would add it, so still I have to add TEMET in the video. I add it when we are ready with everything. It should be small volume, 2 microliters now. According to the protocol, I add it to the main tube. I will mix it and put it in the cassette for the polymerization. So work carefully now with the acrylamide. It's a liquid, so it's a toxic when it is not polymerized, so be careful that it doesn't go into direct contact with the skin. It will be absolutely safe after polymerization, but when it is in solution, it is still toxic component. And when we are ready, carefully with blue tip and 1 milliliter pipette, you add it to the cassette, if necessary twice. So now you see if there is a possibility to remove some bubbles from the surface. So we don't need bubbles also here. You remember bubbles story from yesterday to remove the bubbles from the needle and the syringe for homogenization. We also don't need bubbles here in the gel. So now we carefully insert the comb to be sure that there are no bubbles. And after insertion comb, we leave it for 30 minutes for polymerization. Right? Now we have time, the 30 minutes, to prepare samples. These all our samples for the one gel, as you see, PNS1, S101, P101, so it's from one student, and 3 samples, and the other in total 9 samples from one group. We incubate 5 minutes at 95 degrees, and after this incubation with centrifuge 5 minutes at 13,000 G at the room temperature. So this is how we prepare our samples for electrophoresis. And basically now we start centrifugation, and we will be ready with the sample to start. Yeah, so it reaches 13,000, and I guess it will be much faster now online. Again, the loading scheme in the protocol, and these are equipment. So on the bottom, so I will make a short pause. So this was the plastic like a cassette, which is, so to say, empty. It's a dummy. It's in a case if you have only one gel, we use it. This is the electrodes and the chamber, how we assemble and run electrophoresis, the clamp to fix the gels, and the electrodes which are connected to the power supply. What is very useful is to label your wells before you remove the comb, right? So you label that you can see it later. And extremely important for this gel cassette, plastic, commercial one, to remove this tape before you assemble the system. Yeah, so this tape gives you the gel ending which will contact the electrode buffer, and when you I took this dummy cassette on the other side, and then you insert it into the system. The system is easy because it's only one way. You cannot make mistake left or right. And only one way it goes inside, and then you fix it with this clamp, and we are ready. Right? So when we are ready with that, we fill the chamber with the electrophoresis buffer, which we have prepared. So this is the Lamley buffer, and we fill it in the upper chamber. So this is our upper chamber, which goes into contact with from the side where you have a comp, and how you will load the samples, your protein samples. So now we fill it till the top. And on the other side, so that will be lower chamber of electrophoresis buffer, we just fill it to the level where we had the tape, that this gel has a contact with the electrophoresis or running buffer. So this is the same definition, running electrophoresis or Lamley buffer, and we carefully remove the comp. And now we would be ready to start with loading our sample. Now you see how useful it is to label these wells, because you would not see well how where actually the ending of the well is in the comp. And these are gel loading tips. They have very long and narrow extension here. And just to help us with the loading sample, we take our protein samples. They are blue, and you will see how I load it into the first. So first was the markers, and I load the sample into the first well. And I take all the samples one by one, so probably second would be PNS number 1. As you see, I put back what is left into the rack, and I load it, second sample. Right? You don't have to go completely to the contact with this mark, but at least you see position of the well. The sample is heavy. When you press the pipette, now maybe you will see it, it goes down itself, right? So don't go too down in the well. It's heavy, it has a glycerol, and now you see all the blue samples loaded into all the wells. When we load all 12 samples into the gel, it's ready to start, or we started 2 gels. On the other side, the second gel can be started in parallel, and we start electrophoresis. We agree to start at constant amperage 20 milliamperes, and it slowly will start to increase the voltage here. We put 1.5 hours, and basically this we are ready. So electrophoresis now is running. It's the end of the video, and I think it brings me to the end of the first half of presentation today. Now electrophoresis is running. If you have any questions, please put them into the chat. I will check them now. We have a 10 minutes break, and exactly in 10 minutes we start the second part of our presentation today. If you have questions, please put them into the chat. See you later. So welcome back to our second part of today's online live stream. And we discussed in the first part, and we start the electrophoresis run. This is the end of the movie. I do not see any urgent questions in the chat, so I hope everything was clear for you. And So we basically proceed with the second part of today's presentation. We are done, or basically when electrophoresis is running, usually it's something like 1.5 hours or so, we have basically time to prepare the next step. And next step is protein transferring, Western blotting. And we need a few important things to prepare. Today we will discuss a few theoretical points. It will be a bit shorter, but in the practicum, in the real life practicum, we have this break. If we can have a coffee or the lunch break, depends when we start. What at the moment I cannot offer you, but what we do every year, at least last 4 or 5 years, I think, with the moment students, that we make during the electrophoresis run, we make a SDS-PAGE flash mob. So basically we divide us into several groups. Some students want to be proteins. We find the bigger proteins, the smaller proteins. Some function as the SDS, some as DTT, and some are chloride ions or the glycine ions, and we basically make such a flash mob every year. And I just would like to invite you that you join the flash mob in the 2021. So hopefully we will have possibility, and there will be no corona crisis, that we have a big crowd for the flash mob, that we can maybe make it in Biocenter foyer and make a SDS-PAGE flash mob. We will see. So now this is the break or the running of electrophoresis. Let's discuss and plan our protein transfer. So this is how the list in our protocol book, like what we have on page 18. Again, simple things, equipment we go through now fast. We use the same gel transfer unit. This is basically the same. You will see there are small differences. We use the camera, it's called CCD camera, to detect results of our Western blot. We will need the shakers, we need the membrane. So we use a nitrocellulose membrane. There are different membranes basically which we can use for the Western blotting, and besides nitrocellulose you will probably hear something which is called PVDF. In any case, these are membranes we use for the basically to get a replica from the gel or a replica of protein on the membrane. We need Whatman paper, so thick filter papers of the same size as the gel, some sponge pads, and solutions. So let's go slowly through the solutions. The gel transfer buffer, maybe you remember or you paid attention before. Basically, this is the same Lemley buffer, 25 millimolar trees, the glycine, SDS, and there is a 20% ethanol here mentioned. 2 points I would like to say now. So the SDS is optional. Sometimes SDS is added, and often SDS is not added. This is important because it depends on the proteins basically of interest, that these markers, proteins of interest, what we are going to detect with antibody. For some membrane, for instance, protein markers, components of organelles, membranes, and so on, we add SDS at the same concentration like for electrophoresis. Otherwise, we cannot, the proteins do not move, do not electrotransfer from the gel to the membrane. But often we omit SDS, and then the reason is that SDS can interfere with the binding of proteins to the membrane, to the nitrocellulose membrane. So this is where SDS might interfere, but you will see it in the future. In the practicum, we use SDS. Ethanol here, 20%, is the alcohol we use. We add to this more or less Lemley buffer. More standard and routinely in our laboratory we add methanol. For the practicum, for the safety regulation reasons, we add ethanol because we can use it normally. With methanol, the regulations are quite strict and we have to be more careful. But basically, these are details. What is important that we add alcohol, methanol, or ethanol to keep the gel during the transfer procedure. So basically, when we take the gel from the cassette after electrophoresis, that it stays in the same size during electrotransferring. Right? So this is the main idea. And second, as important, function of the methanol or ethanol that it's facilitate binding of the proteins to the membrane, to nitrocellulose or PVDF membrane. So these are function of the alcohol here. The blocking buffer we use in the Praktikum is 3% BSA, bovine serum albumin, 1 millimolar EDTA, twin, and sodium azide. So let's go briefly through the components. This is the protein. It can be any other protein, but this is available so that we have it in the large amount. And obviously the reason is clear. So it's to block the membrane, to avoid unspecific binding of the antibodies to the membrane. And this is basically the same reason as you have discussed with Ilya 2 days ago for the immunofluorescence, that you use the blocking buffer. So for this, we use as a blocking, we use BSA, but this would be the same function. Or here you see for the secondary antibody, milk powder would also function as the blocking component. Just the difference is that in milk, the main blocking function or main protein is casein, as you may know, and the milk is a component of thousands of proteins, so often the milk is a better blocking agent. But the main reason for you to understand is that we want to avoid the unspecific protein binding. Yeah, so to put it as down as little as possible. A DTA function also to avoid the unspecific protein-protein interaction. Twin 20 is weak detergent, which also function as the to reduce the unspecific protein-protein interactions, and we use a low concentration of twin 20 for this reason. And the last component here is sodium azide. It's antibacterial, so the toxic compound, which when we prepare the blocking buffer in advance, that it can stay for a longer time. We add it to keep it, not to prepare basically every day. So the washing buffer you will see on the procedure on the protocol is the Tris buffer and the sodium chloride, again a bit of the twin 20, to wash many times in different steps to wash out different antibodies, non-bound proteins. So these we use for the washing, and you will see we also use the secondary antibody, again in a similar for like it was in immunofluorescence, but label and detection method will be different. And usually we have a secondary antibody in this type of buffer, which contain milk powder in washing buffer, but the function of this milk is the same as the BSA in the blocking buffer, to avoid unspecific protein-protein interaction. And I will explain you what this ECA enhanced chemiluminescent substrate is in the next slides. So basically, these were components of the material and buffers for the Western blotting, and now we come directly to the video tutorial that I will click, and you will see how to proceed. This was our ending of the electrophoresis, or the starting, and these are components. So let me put it a little bit back, it was too fast. So these are what we need for the before we start protein transferring procedure. So this is what we will use, this special kind of spatula, large. We'll use it to open cassettes, and these are the electrodes, 2 of them. You will see it later for the protein transferring chamber. Then we see the container for the protein transfer buffer, or the transfer buffer. Yeah, exactly. So this is transfer buffer, I explained in the previous slide. These are sponges we use, and the tube. You will see the reason why we need the tube. So these are Whatnot paper, thick Whatnot paper, and the nitrocellulose membrane. Usually it is between these blue papers to keep it. It's more to protect. It's a bit breakable, the nitrocellulose membrane. and we need this. We need the forceps. So what else is on the table? Right, we are ready. We pour the transfer buffer into this container that we pre-wet all these sponge pads and filter papers and nitrocellulose membrane in the transfer buffer. In the case in the future you will use PVDF membrane, you will need to activate it. It's called to incubate it in ethanol or methanol. We use nitrocellulose. We don't need this activation of the membrane, so we pre-wet everything, sponges and the Whatman papers and the nitrocellulose paper. So carefully taking it from this between blue storage, so to say, papers and put it in the buffer. Could be a little bit more, but it's okay. And we proceed with the sandwich assembly. So we take this anodic plate, you see. So I will take a pose, make a pose. You see this plate. So this is electrode. Yeah. So you see this is everything metal electrode. Yeah. Often it is made from the platinum actually. And this is the second. This is cathode plate. So it's opposite to the tiny electrodes in electrophoresis. This is the whole plate, and actually for the different approaches, like what we, for instance, use so-called semi-dry, this plate can be really huge, like 20 to 30 centimeters or so large. So this is for one mini gel, or actually two membranes can be assembled here. So this is the anode plate, and we now assemble sponge pads and the filter papers. So we take these sponges, two of them, and assemble it in a way like you see. Right? So this is the first sponge, now the second sponge, and the Whatman paper. Carefully, slowly, no pressure, no hurry. And now we go to our gel. I would have to explain for this video, electrophoresis was running just 5 minutes. So normally you see the blue color. So we loaded the sample and I kind of pretend that electrophoresis is over, but the blue runs to this level, right? So this will be the end of electrophoresis. Just for this video tutorial, I stopped electrophoresis from a few minutes after the start, but even here you see the concentrating effect, yeah? So we can come back to my explanation how the proteins are concentrated by this method of a Lemley, and we load it like large volume, and now it's already starting concentrating, although it even didn't reach, it's come just closer to the resolving gel. So we pretend that blue is now entering or leaving actually the gel, and what we have to do is to remove the gel from this electrophoresis cassette. So we use this large spatula basically to cut, because it's like angled, it's polymerized, and then it's 90 degrees on this direction. So this we have to cut completely and carefully. So this is what I'm doing. I'm cutting this ending of the gel. We don't need that. And it will interfere with the assembling of the sandwich for the protein transferring. So we have to cut it carefully. Now we have to disassemble this cassette. So you see it's only for one use cassette. You cannot reuse this, and we disassemble this, the upper and the lower plate. Now you basically have to go on all the sides. Behold, this has these plastic contacts. We have to disassemble them, and when it's disassembled on all the sides, you take it, lift it up, the cassette, and you never know where your gel is. Often it comes in the upper or in the lower. Now we have it in the lower cassette, and you label. So first we remove the stacking gel. We don't need that. So sometimes we leave it, but usually 99% of all experiments we remove the stacking gel because it's low percentage. You just make a dirt and the smear. You cut your one angle that you know how you load your sample, so this is up to you. You can cut left or right. And when we are ready, or just of course if you cut it, you should remember which side. In our case, it's easier because we have, so the markers we see, you carefully assemble the nitrocellulose membrane on the top of the Whatman paper, so now it is ready, that you don't have any bubbles and so on. You can add a little bit of the transferring buffer on the top, and now we take the gel. So we carefully lift up the gel. It's also a bit tricky in this system, but again, it's just for this system, but carefully take it in the hands and the left and right corner, lift it up and position nicely on the nice nitrocellulose membrane, right? Usually it's one move. You put it down and it's positioned nicely, center it. On the top of the gel goes Botman paper and then sponges. So now removing air bubbles. This is important steps. We use this 15 milliliter tube and you roll the bubbles out. Potential bubbles. I hope I didn't have any bubbles, but in the case if there are bubbles, we remove them by rolling them out from the gel. Basically, from center you roll it on the side. From center you roll it on the side. Then you put 2 sponges and you assemble cathodic plate on the top. Carefully, so it's also one move. It goes on the top, and you take this system carefully with your hands, like I do, more or less. And it's one way that you do not make mistake, other way it will not go. You assemble it back and fix it with the clamp, right? It's fixed, and then we add transferring buffer just to this cassette. So it's very low volume of the transferring buffer, and it will go only between in this inner chamber, so to say. It's small volume, it's maybe 50 milliliters or so in total, and actually even in the company protocol book you can read that it's called semi-wet method. It's just something in between of original wet transferring system and the semi-dry Western blotting, so they call it semi-wet. But important is for you that you feel this chamber where you have a gel and membrane, your sandwich and sponges, everything, just on the level of sponges. So basically you cover your gel, you cover your nitrocellulose membrane, and this is enough. You don't have to fill it till the top. And the reason is that if it's too high, then basically the electrodes will go not through the gel, but just in the buffer. You don't need that. So you are ready now, and you, after filling of this chamber, so what is also good is to fill on the outer chamber here. That's why I actually wanted to explain you, because it is not in the video. You fill the surroundings, so the outer chamber with ice, just to cool the system, because actually it produces heat, and then to keep the temperature under control, we fill it with ice. So now I jumped back to the movie again. Again, we fill our inner chamber with the transferring buffer and connect electrodes. It's also one way, so red to red, black to black, and we start transferring, that you will not make mistake. 100 milliamperes, 90 minutes, it's absolutely fine. So after 1.5 hours, we will have our transferring done. So 1 hour is over, and this is how examples, basically how our results will look like in the Praktikum. So, and how we proceed, we will disassemble all these chambers. So we take out our membranes. So this is the photo of one of the membranes from the one group. And you will see on the membrane, since we use these pre-stained markers, so this is a photo from the company, how these markers look like. They are pre-stained in a way that, so as you see, we have a ladder from 10 to 170 kilodalton. And 2 of these markers are, all of them are blue, but the smallest is yellow, 10 kilodalton, and 70 kilodalton is red. It's somehow coincidence, it's good for us, because when we get a membrane, it looks like that in black and white photo. But what we do first, we wash a membrane in the water for some short, a few minutes, 2 minutes or so, and then we cut it for us, for our experiment, into 2 pieces. So one of our marker we want to detect is in the upper membrane, and the second marker is in a lower membrane. So we basically cut in a level of 70 kilodalton on a red marker, and you will get, each group will get such 2 pieces of the membrane. Before cutting, the membrane will look like that. Yeah? So you have on the side your markers. These are 70 kilodalton, not necessarily exactly in this resolution. This was just an example, and I think the percentage of this gel I made a photo of is higher, because so in your case the red marker will be somewhere a bit lower, somewhere in the middle more or less. So but important is that you cut your membrane in the level of the red marker, 70 kilodalton. The upper will go for the one antibody, and the lower membrane will go for the incubation with the another antibody. And you can see here the 10 kilodalton 10 kilodalton marker is yellow, and all the rest are blue, and one of them, 70 kilodalton, is red. So this is how your membrane will look like in the practicum. I would like to give you just an example of the membrane of my experiment, probably 2-3 weeks ago or something, when I was making this presentation for this online course. I made a photo of my membrane where we use not pre-stained but unstained markers, and we load them on both sides, on the left and right side of the membrane, that cutting can be more careful. That, for instance, these are different masses here. They are not the same as the pre-stained markers, but you can see the principle is the same. So you have low molecular weight, like 10 kilodalton, and some markers are, for instance, this is 25, this is 50, and so on and so on. You have the proteins transferred from the gel to the membrane. So your membrane look like that. Of course, this membrane was stained with the protein dye. In this case, it's amino black, for instance, and the membrane look like that. Yeah, in the practicum, you will have membrane like that. We will not stain the membrane, but we can. So normally, we have this membrane stained with amino black, for instance, but there are other dyes like punso red, for instance. Amino black is quite common. So basically you see on the left and right side markers, and if you want to cut your membrane carefully, for instance, just below 25, you just nicely cut it below 25, and so on and so on. Often when we do this in laboratory, Western blot, we can cut the membrane like in 3 or 4 pieces. Then you have certain markers above and below that you know exactly the position of the molecular weight of band of interest you want to detect with Western blotting. So the next step will be, so after cutting, and so washing and the cutting will be to label your molecular weights with a pen, so normal pen, like here it is shown here, like in this photo from the last year students. The positions are labeled like 25, 35, 40, and so on and so on. So corresponding to this example on the markers on the left side. So this is how your membrane will look like, cut and labeled molecular weights, and number of the group, like for instance, I think it's labeled group C or just C, right? So let's proceed to our protocol book, and this is just a list of the steps what we are going to do with you after transferring. The next step after cutting and the labeling molecular weights marker, we block membrane in the blocking buffer for 1 hour. We already discussed the components. So this blocking buffer contains BSA, and we want to block the membrane with protein and the twin-20 to avoid unspecific binding of the proteins, so the antibodies to the membrane and to the proteins which we have transferred from the gel. So this is the reason for the blocking. 1 hour at room temperature is standard approach before we start the blocking with primer antibody. So in our protocol for practicum, we incubate our membranes in the overnight with the primer antibody, and these primer antibody are diluted in the blocking buffer, the same as we have used for blocking membranes. And this is one of the approaches, that incubation overnight at 4 degrees. So we use the cold room for this, where we have shakers. Alternatively, this can be shortened to a few, 2-3 hours at room temperature, depends on the situation we are. So upper part of the membrane will be incubated with anti-transferrin receptor antibody, and the lower membrane will be incubated with anti-tubulin antibody solution overnight. So the next day, morning, so when we start the practicum, we rinse the membrane 4 times in the washing buffer to wash out the leftovers or the not bound primary antibody solution. So we do this carefully 4 times in excess of the washing buffer, and then we start with incubation with secondary antibody. So 30 minutes at room temperature is really minimum. Often we do this 1 hour. So for our markers protein it should work. So 30 minutes incubation with secondary antibodies. And then again washing, extensive washing 4 times 10 minutes in the washing buffer. Draining of the washing buffer and incubation that we don't have some kind of leftovers, just maybe a few drops, and incubation in so-called ECL substrate for 2 minutes. So these are in total 9 steps. And last one, enhanced hemiluminescence substrate for 2 minutes for detection of luminescence. I will explain to you on this slide. So this is hemiluminescence reaction, what we perform and how we detect signal in the Western blotting, as an example. So actually this is what we will do during our practicum. This, what I have illustrated, like the light blue is our membrane, and all the proteins from post-nuclear supernatant, from all the samples, cytosol and the total membranes are transferred to the membrane. And this is kind of the way I illustrated low molecular weight protein up to the high molecular weight proteins. And for instance, this is our marker proteins, but of course on the membrane there are several thousand or 10,000 proteins. In the same sample we load, we want to see only one specific protein out of 10,000 proteins on the membrane, and we can detect it with a good antibody, good primary antibody, which can recognize only one specific protein of interest. In our case, it's a transferrin receptor or the tubulin, for instance. So primary antibodies are immunoglobulins. Normally they are either monoclonal, mouse monoclonal, or rabbit polyclonal antibodies. This is important to know species specificity. Is it mouse or rabbit? Of course, they can be different species, but this is most common. And we incubate, as we discussed, usually overnight with this primary antibody solution. That we have this antibody bound just to the specific protein of interest, our marker. The rest will be washed out with the washing buffer and washing steps. So the second incubation step is a secondary antibody. What we need to know, what was the primary? Is it the mouse or the rabbit? Just an example. So we need to use the secondary antibody, anti-mouse or anti-rabbit specifically, right? So we also incubated for some time, 30 minutes or 60 minutes, and we have this kind of the sandwich membrane, the protein, primary antibody, secondary antibody. So all the proteins are here like bound to each other specifically, hopefully. But what we actually detect usually, and also majority still as a routine, as a practice in the laboratory, we detect actually enzyme. And this enzyme, in this case, you might know it from before, is HRP. It stands for horseradish peroxidase. So the secondary antibody is covalently bound to the enzyme HRP, and this is what we detect, for instance, in this chemiluminescence reaction. So we detect enzymatic activity. So the more antibody, the more we have the protein on the membrane, the more antibody primary binds, the more secondary antibody binds, and we can enhance the signal in enzymatic reaction to almost unlimited or very, very, very strong signal. What we want to see is a signal we can detect with these sensitive techniques like what we have in the CCD cameras or the films that we detect the signal. So what do we do with you during the practicum and also routinely in the laboratory? We perform the hemiluminescence reaction, which is reaction for the peroxidase activity. This is an enzyme which always takes two substrates. One is the broad, very broad specificity substrate number 1, and we use luminol in our case for this reaction. The second is always hydrogen peroxide. So these two substrates are taken by enzyme peroxidase, HRP, and we get the products. There is not really need now to discuss what kind of the products. It comes here in our case. What is important besides the product of the reaction of the luminol here, that's why I wrote the product, is a light. So this is the light, 425 nanometers, we can detect as a result and the readout of this chemiluminescent detection, and we detect it either with films, sensitive films, or with the cameras, CCD cameras in the laboratory. So this is what we detect, light as the final readout of all these, so enzymatic reaction and all this procedure, what we have done. So that's what we detect is a luminescence. And this is summarized in the last slide. For this procedure after transfer luminescent detection, so we, after incubation with reagent, with the ECL substrate, we remove the substrate, so drain basically the last drop, and we expose the membrane for different time, for instance, seconds or minutes to 15-20 minutes. In some of the ECL substrate, especially commercial, they are designed for a very long signal that we can detect very weak signal, original on the membrane that even lasts for hours. On the CCD camera, how we detect the signal, and usually it's different time points, just an example, 1, 3, 5, 15 minutes. And we save our images, what we get. We make a photo of the markers, what you have made with a normal pen, and we overlay the signal of the Western blot in the computer, which come from the luminescent detection, and the markers, label them, so the images nicely as a final results. An example I'm going to show to you today, I believe it's the last slide for this part of the presentation, and what I would like to show you are results from the last year practical course, and basically these are antitubulin and on the left side, and the antitransferrin receptor on the right side, and these are different groups. You can even see the group E, G, B, D as an example, completely random, and 3 different students in each group loaded their samples. So sample number 1, sample number 2, sample number 3, and this is how we detect how the signal look like now when we save the images with antitubulin antibodies and antitransferrin receptor antibodies. So you can see what is important here, the molecular weight, what you have labeled, or other students last year labeled on the images, and the overlay of these markers, and the signal from Western blot chemiluminescence detection. So this is for the transferrin receptor. And what you can see that so again, these are 3 different students from the one group. So you will get these results for you as a homework. Now you see in total, let's say 4, so 12 students result. You see they are very highly reproducible, the quality is very good. So take your time, you have it already in Elias uploaded example of this result, you see it now. Take your time to think. You will get also additional homework file with example, one of these results from one of the groups, to analyze where you have, in which sample you have the signal for tubulin, for the transferrin receptors, and analyze which sample might correspond to the postnuclear supernatant or total membrane fraction or the cytosol. Think how our markers might be distributed. So this is what might be distributed. So this is your homework to analyze these results of our Western blot and exemplify it from the last year practical course, find out the order of fractions loaded, and at the end to draw conclusion on distribution of these 2 markers between two subcellular fractions which we have purified with you during the subcellular fractionation experiment number 2. I think I am at the end of the experiment number 2. We still have enough time for the last topic of today. If you would have questions, I hope that everything was clear, but if you have questions, please ask them now, that I will see them in the chat. If not, you can always ask it later. We will have time after live stream today or emails. So basically, this is just an example for you and as homework to think what result we get, analyze them carefully. Sometimes so you don't have a signal. Some students have it completely, no signal in certain fraction, a little bit in other students, so triplicate from the one student, triplicate from the second student, and triplicate from the third. So think about all of that and try to make your conclusions. So I will change the completely gears now and come to experiment number 3, which is WST1 cytotoxicity assay. There will be just a few slides. This is a small experiment we are going to do with you, but it is important. Important is for us, for the practical course, because what we measure and what we want to know is in this cytotoxicity assay is the concentrations of the compounds we use during practical course. You remember that what you got introduction in the very first day on Monday from Mariana, that we use Velcade and Taxol as the two compounds basically during for all our experiments, for IFs, for the proliferations assay, and here in the cytotoxicity assay, what we want to know is the active concentrations of these compounds, but also any compounds we use for similar experiment. And this is the way how to do that. So you perform cytotoxicity assay, it's called WST, and the principle is rather simple. So these tetrazolium salts, and the name is water-soluble tetrazolium, it stands for WST, is cleaved to soluble formazan. So this is the product of the reaction, which we will read in this assay as a readout, and this is cleaved by the system, the enzymatic system, mitochondrial dehydrogenases. So this succinate tetrazolium reductase system and the name of this enzyme, and this is a part of respiratory chain of the mitochondria. This respiratory chain is labeled here as RS, and the WST, the chemical structure, is here on the left side. So basically, what you don't need to know all the details. There are some electron coupling system here. It's not really so important because it's always different. These are commercial kits, and what I would ask you to remember that one of these tetrazolium salts, in our case, is WST, but you will probably hear and use in the future different ones. I saw commercial WST8 already, and maybe even higher, and the old ones are XTT or MTT. They are all tetrazolium salts. So from something what you add to the cells, if cells are alive and mitochondrial respiratory chain and dehydrogenases are active, you get the product NADH, and the enzymes cleave the reagents like WST, and this product, the formazan, we read at 440 nanometers in the 96 well plate and 96 well plate reader, the same what we have done yesterday in the Bradford assay. So basically the principle is simple. If cells are alive, we have the product. If cells are dead because of cytotoxicity of certain compound, we do not have this product, and this is a little bit light red and this is brown red. We can read at 440 nanometers. So this is how it looked like in our Praktikum protocol. We have 96 well plate. Most probably you will get it ready from our technicians. If not, then you will split from the 10 centimeter dish to the 96 well plate, and this will look like that. After growing 1 day after trypsinization splitting in 96 well plate, we will add Taxol and Velcade, and you will get stock solutions, 10 microliters of the Taxol, 1 millimolar and 1 millimolar Velcade. You would have to make dilutions 1 to 10 and quantify the concentrations of the Taxol and Velcade, starting from the 10 micromolar, I believe, down to the low range. And we need the EPIs and the multi-channel pipette, the same as we have used for the Bradford assay, and the reader you have used already before. So in the procedure, we start with the seeding, so 5,000 cells per well in 100 microliters. Next day, you will prepare these inhibitors dilutions. And meanwhile, when we prepare inhibitor dilutions, I would like to ask you the question that you stay focused and think a little bit. If you know what IC50 stands for, I would like to write it to the chat. IC50. This is what we get as a result of this cytotoxicity assay. And not only this one, but often the activity of many chemicals, many compounds, many inhibitors, many drugs which you use, if they are tested in vitro cell culture, we come with the numbers or with the concentrations what is IC50. So if you know what is that, please write it to the chat and I will keep you guiding through this protocol. It is rather simple. I didn't make a video for you for that because everything what you should know you already know. So basically after you are ready with the dilutions of the chemicals from the 10 micromolar down to the nanomolar range, you aspirate medium and add 100 microliters per well in triplicate, the different concentration of inhibitors, and incubate it for 48 hours in normal incubator. So 48 hours, it's 2 days. After 2 days growing, so I think it is, we can make it shorter, of course, 24 hours, but we will see effect of the inhibition, for instance, of cell growth. What you might think about, if you know already and you remember the introduction by Mariana and in the first day, so that sometimes we can see inhibition of cell growth, and if you have the duplication rate of the 12 hours or 24 hours, depending on cell line, depends, and you can expect what you see. And basically 48 hours, this is what we decided to have with you during the practicum. After this, you aspirate medium and you add 100 microliters again per well of this WST reagent, diluted 1 to 10 in the cell culture medium with the multi-channel pipette. And you can add this reagent in the empty wells as the background control, and we incubate this mixture 1 hour at 37 degrees that cells can take WST reagent and metabolize it to this compound we discussed in the previous, so the formazan. So 1 hour is enough that we can already read our results. After 1 hour, we read it as absorbance at 440 nanometers, and after this, we will prepare our graphs of the results and calculate these IC50 for all the inhibitors we are testing. I see something is coming in the chats. So Moritz wrote, it tells you the concentration of the substance at which it reaches 50% of its inhibitory effect. Michelle, IC50 is the concentration of an inhibitor that is required to block 50% of a target, for example, enzymes or receptors in vitro. It is the half maximum inhibitory concentration, Elias wrote. I think you all are right. IC50 stands for the half maximal inhibitory concentration. And this is the quantitative measure that indicates how much of the specific substance we use in our test to inhibit any of the biological process we are interested in. It can be cell growth, it can be enzymatic activity, and so on and so on. Thanks for your activity in the chat. And basically, I believe, at the end of today's live stream. We know now all what IC50 stands for. I give you example from the last year, again, the students, one of the probably students or the group, the results of WST assay, 48 hours, and it was actually 90 minutes WST, not 60 like it was in the protocol. And you see results. So when you have a low concentration, 0 at the low concentrations. So here we have micromolar of Taxol and Velcade, Taxol blue, Velcade red. And here we have optical density 450 in this case. And basically these are graphs. They are similar, not exactly the same, but similar. Then when we have a low concentration, that there is no toxic effect, so basically cells do not see it or ignore or metabolize it, that it doesn't cause any cytotoxic effect up to certain concentration of the chemicals, which would be here, if I see it correct, would be 1 nanomolar range. It doesn't work, and suddenly within the, well, I would not say it's a narrow concentration, but this is between 1 nanomolar and 100 micromolar. So here you have no effect, then we see effect, and basically in 100 nanomolar concentration range, the cells are basically dead, because in 1 micromolar and 10 micromolar they are as dead as here. So basically you see the range in 2 orders of magnitude. Well, it's quite large, quite big range, 2 orders of magnitude, that this activity of these chemicals you can detect. And please, so now you know what is IC50. You have a graphs as an example. Your homework will be to analyze these graphs, what you see, what are parameters on the x and y, and where is, for instance, Taxol and Velcade. Not necessarily in your practical course you will get exactly the same graphs, but they will probably similar. So estimate rough IC50 for Taxol and Velcade. You don't have raw numbers. I will not provide you Excel sheets with all the values. So estimate what you can expect as IC50 in the range. So range nanomolar, 10 nanomolar, 100 nanomolars, micromolar, analyze it and estimate the rough range. And what is important, what I would expect from you, so the values you get as the approximate concentration of IC50 for Taxol and Velcade correlated with the values which we have used in experiment number 1. You have used it already online with Ilya for IFs. And tomorrow you will have another online stream with experiment number 4 with Mariana. So correlate this value what we got now with WST assay with the real concentration we use for the biological experiments also during the practicum. So this will be your homework, and I am at the end of my presentation today speaking about homework. You will get an explanation tomorrow at the end of our live stream with all the details about homework again. Summarized as one file in one piece. What is important, you have this already, this file uploaded in Elias. And that besides that you have this file already, you can see, maybe you can see it, take a look at this file today, but tomorrow Professor Huber will explain again everything slide by slide, what homework is for you and what we expect from you after this live streaming and this cell biology course online, so that you will get all the instructions. So I think I am at the end, and I would like to thank you for your attention. We will stay in live stream for another 10 minutes or so. So if you have questions, please write them to the chat. Otherwise, thanks again and all the best. Ciao.